Good afternoon. I hope I didn't scare too many people off. It's a bit emptier than it was before lunch. I'm Olav Hellebo. I joined ReNeuron a couple of months ago, and I'm from the immunology oncology space. So I also thought about what can we learn about dendrion and provenge was a question earlier that I think we probably didn't address. And coming from outside really this space of regenerative medicine, I think it's important to look at that, what we can learn from that. And one of the things is cost of goods and value. Those two things have to hang together. And I think for provenge, they didn't quite hang together. I noticed in their latest quarterly report, they had 49% cost of goods, which is a lot when you're selling $50 million a quarter, or was it 50 million pounds? So, I think that was one issue. Another one was expectations. I mean, the expectations around provenge were enormous. And maybe you can learn something about humbleness as well from that. So for me, kind of two things. One is I like allogenic therapy. I think that has the potential to treat a lot more patients, and that you can be able to deliver enough scale to those patients. So it's a product rather than a process. But let's get into the presentation and talk about ReNeuron. I think we have a lot to be humble about. Disclaimer, first of all, spot the typo. None, huh? So ReNeuron, we are a clinical stage company in the cell therapy space. We have allogenic stem cells. We're using a highly scalable technology, and yes, we're getting great help from Catapult to make sure that we can do that right. And we are in phase 2 for stroke. We are in phase 1 for critical limb ischemia, CLI. And we're also in the late preclinical stages for cell line targeting ophthalmic diseases. Our CSO, John Sinton, what really gets him up in the morning now is actually kind of the next thing, which is exosomes. So we are able to extract exosomes during our manufacturing. And exosomes are about the vesicular transformation of information between cells. It's very much an emerging area of biotechnology, and we're very delighted to be part of that. And I'll come back to that a little bit later on. We have strong investor backing, which is important. And we are building a manufacturing facility right now in Wales with the help of government grants. And coming again from the antibody space, you know that manufacturing is really a key. Not only to control your costs, but really to control your knowledge, your know-how. It is very much a key. So far we're using CMOs, but in the future we will have our own dedicated facility in Wales, and I think that's an important part also for us. So our technology platform consists of 2 cell lines, and we are investigating those 2 cell lines in 3 different indications. The CTX cell line was derived from neural tissue about 10 years ago. A single neural stem cell was selected, conditionally immortalized and cloned. And these CTX cells are now in 2 indications. And as I mentioned, we're extracting exosomes during the process also at the same time. The retinal cell line is human retinal progenitor cells. They're derived from fetal retinal tissue. So it's not a clonal cell line. This is a cell population expansion. We're using a patented low oxygen culture to do this. And we have in-licensed that from Dr. Michael Young's lab at Shepard's Eye Research Institute at Harvard. So our platform. Since the CTX cell line is derived from a single cell, that means every cell is the same. That gives us at least an opportunity to make sure that we have a consistent output from manufacturing. It makes us also scalable and cost efficient, particularly obviously at a high scale. We deliver the cells directly to the target site. And since every cell has the same potency, that gives us a total dose that is relatively lower than it otherwise would be. Last year, we were able to develop a cryopreserved drug product. And that was a real breakthrough for us. Logistics is difficult when you run clinical trials with short shelf life, and we're able now to have 3-month shelf life on these products, and that has been very well received by our sites doing our clinical studies. A couple of words about exosomes. So there's extensive protein and genetic information sharing between cells, and exosomes are a vital part of that. So these exosomes are nano-sized vehicles secreted by stem cells, and they contain a multitude of RNAs, cytokines, and other important proteins. So far, exosomes have been used as diagnostic tools, and we are looking into the therapeutic potential. Right now, we're looking preclinically at wound healing, immunology, and cancer, and we will be selecting our first indication soon. So here's a snapshot of our pipeline. We are in phase 2 for stroke. The trial is ongoing across 9 sites in the UK. We have started the phase 1 in CLI. It's the same cell line as in stroke, so the CTX cells. And then we are conducting preclinical studies in retinitis pigmentosa, which is disease causing blindness. So we're looking at, in addition to that, the CTX cells on the exosomes from that preclinically. Let's dive into stroke a little bit. In the US alone, there are 800,000 cases of stroke every year. Europe and Japan is an additional 1.4 million episodes of stroke per year. So this is a huge market. Japan probably being the one that's growing the fastest. They have a very fast increasing aging population in Japan, and stroke, ischemic stroke, is a big issue there. And that might be the reason why they decided to change the regulatory system for regenerative medicine, because they know they have a big issue with stroke coming over the next decades, and they need to do something about it. And it's kind of interesting to see a government actually, for once, thinking about the future and doing something about it. You know, it's innovation, isn't it? The outcome for stroke victims range from death to spontaneous recovery. About a third will be left with chronic consequences such as paralysis, and this obviously has a large social and economic burden. The pharmaceutical treatment window right now is in the acute phase, so the first 24, 48 hours. We are looking at the chronic phase, so a few days or a few weeks after stroke is what we're looking at. Athesys will be presenting, I think, later on this afternoon, and they're dealing with the acute phase. And they have a phase 2 trial ongoing there, and we're all crossing our fingers that that's going to be a success. The results are coming very soon, and that's the one thing we need in our field is good clinical data. We're talking a lot about technology, but the clinical data is really what we need. So back to CTX. Our clinical models have shown that our stem cells induce growth of new neurons and blood vessels, as well as triggering the immune system to reduce inflammation. So let's look at the clinical data. We have conducted a phase 1 dose escalation study in 11 patients. They all suffered from moderate to severe ischemic stroke, and this procedure was performed on average 2 years post their stroke episode. The stem cells were delivered via standard stereotactical surgical procedure. Patients spent 1 or 2 nights in hospital for this. No adverse events attributed to the stem cells, and there were no immunological adverse events either. This was a safety study, but obviously we did have a look at efficacy, as you do, when you can. And we noticed consistent improvement across all several different efficacy scales, both at 3 months post-procedure and 12 months post-procedure. So based on that, that gave us the motivation to go into a phase 2 study. We made some changes between phase 1 and phase 2. The biggest one is probably that we're treating patients now closer to their stroke episode than we did in phase 1. So in phase 1, we did it 2 years post-stroke. Now we're going to about 2 to 3 months post-stroke. The reason for that is that we believe the treatment window, if you can get closer to stroke, there will be less scarring, but you need to be far enough away that the disease is stabilized. So, we're trying to find the right window to do that. The study here is a Simon two-stage study. So we're doing first 21 patients. We need 2 or more of those patients to reach our efficacy measure, which is called ARAT, which is an action research arm test, basically a test where the patient has to grab an object and lift it and put it on the shelf. And these patients obviously can't do that at the beginning of the study. If we reach the 2 patients in the first 21, then we will continue the study to 41 patients, and our goal is to have 5 or more out of the 41. So over to CLI. To me, at least, there is a surprising large number of amputations every year caused by CLI, most of them in diabetics. Any treatment that can increase the blood flow in the lower limbs to reduce the number of amputations would be very, very important. There's been benefits already shown with autologous treatments in CLI, typically from academic institutions. There's been some issues around the quality of the cells and also the time to prepare the cells. So what we're trying to do here is really to build and improve on what's been done previously, but using an off-the-shelf allogenic approach. So we have kicked off a phase 1 study in the UK in 9 patients. So it's a pure safety study, dose escalation, and we'll get the results first half next year. And if there are no surprises, we will kick off a phase 2 study by the end of next year, and that will be a randomized control study. The third program, retinitis pigmentosa, so a degenerative eye disease that can lead to blindness. We have developed human retinal progenitor cells that we've tested in various animal models. The preclinical data are exciting. They show prevention of loss of visual acuity in the RCS rat. And we also see allogenic engraftment as cells migrate into the photoreceptor layer in pigs' eyes. So this is very exciting to us. We may both protect and replace photoreceptor cells. So we're finalizing this work. We're aiming for an IND early next year. So this is what we aim to deliver. Starting from the bottom of the chart, stroke, phase 2 has started, with data from the first cohort expected next year. CLI, phase 1 has started, phase 2 to start next year. The retinal program, we're aiming for an IND early 2015, and a phase 1 to start by the end of the year. And exosomes, our aim is to get in the clinic in 2016. So it's a very exciting pipeline. So in summary, we have an allogenic platform consisting of 2 cell lines being investigated in 3 indications. We're well-backed and well-funded. We raised over $40 million in equity last year and $20 million in government grants. We're very excited about the clinical advances that we have made so far, and we're also excited about the research into exosomes. So thank you very much for your attention.